All right. Hello, everyone. My name is Claudia Lacroix, and today I'm going to be talking, and I'm a fourth year undergrad here in Canada at the University of Toronto. And so today I'm going to be talking about my work on turtle sociality and how snapping turtle vocalizations are associated with energetic benefits. And so the study of social behavior is often neglected in non-avian reptiles. And the reason for this is because they are often less conspicuous than mammals and birds. And so the diversity and complexity of social behavior in reptiles has been underestimated and largely understudied. And despite this, reptile social systems can be quite elaborate, where they exhibit courtship displays, aggregations, parental care, and cooperation. And so communication between individuals also involve visual, chemosensory, tactile, and acoustic signals. Crocodilians in particular have quite elaborate social behaviors, and associated with those social behaviors is the communication and use of acoustic signals. And so a speculated sister group to crocodilians are turtles, and they also use acoustic signals. But our understanding of the function of these signals is largely in their infancy. And so in particular, turtles are a group that were historically believed to be voiceless. But we know now that there are more than 45 species who vocalize, yet 29 of those species are tortoises or terrestrial. And so in aquatic turtles, we still don't know much about why turtles vocalize. And so recently, in the past 5 years or so, more and more studies have recorded vocalizations in turtle hatchlings. And there are approximately 3 hypotheses as to why hatchlings may vocalize within the nest. There's parental care, which has been demonstrated in Podocnemus expansa, which is a South American river turtle. And then there's also the synchronous hatching, where individuals are able to modify development and hatch at the same time as conspecifics, and then synchronous emergence, where conspecifics are able to coordinate and emerge out of the nest at the same time. And so in my talk today, I'll be focusing on the latter 2 hypotheses, where we wanted to investigate whether vocalizations are a feasible mechanism for synchronous hatching and or emergence. And so this was done by performing 3 separate experiments, which I will walk us through today. And we used the widely distributed snapping turtle, Chelydra serpentina, and so the first question was, we wanted to know whether snapping turtles actually vocalize in the nest, which is something that we didn't know at the time. And second of all, if they did vocalize, we wanted to know what hatching period this was occurring. And so to do this, we collected eggs from the Algonquin Wildlife Research Station late in development and set up a simulated nest in a glass aquarium. And so as shown in the picture, we buried eggs in sand with a microphone and left a viewing panel on the side of the tank. We then set up a red light, placed a camera in front of the aquarium, and covered the tank the setup with a box to emit natural light. Then we connected the recorder to the microphone and recorded audio continuously until all hatchlings emerged. So using pictures from the camera, we were able to define 3 hatching periods. The first panel shows the pre-pipping stage, where all eggs have not hatched. The second panel shows the pipping stage, where hatchlings have broken the eggshell but have not started to climb up. And the third panel shows the emerging stage, where at least one hatchling has started to ascend towards the surface. And overall, we found that after sampling 9 hours of recordings, we detected 261 vocalizations and characterized 5 distinct types. To analyze the vocalizations, we used a sound analysis software, and this was called Raven Pro. And in the following figure, we have an oscillogram on the top, and this shows the sound's amplitude, and a spectrogram on the bottom panel, which shows the frequency of each sound. And so type 1 sounds are relatively short sounds with 1 to 3 pulses at a relatively high frequency. Then type 2 sounds are short harmonic or non-harmonic sounds that have 4 to 7 pulses with a relatively lower frequency. Then type 3 sounds are longer with 10 or more pulses and relatively higher frequency. And then type 4 sounds are almost chirp-like with a higher frequency, and these clearly defined harmonic bands as shown in the spectrogram. And then similar to type 4, type 5 sounds are relatively longer sounds that have these well-defined harmonic bands as well, and a much lower frequency and amplitude than the other vocalizations. And so by comparing vocalization count over each behavioral period, we observed that vocalizations were more frequent and more complex after hatching from the egg. So before pipping, we only see type 1 vocalizations being produced, and in the latter half, we only see the more complex types, the types 2 to 5 being produced. So what does this tell us? Well, it tells us that more complex information may be exchanged among siblings during pipping and emergence. However, this is not enough to attribute a communicative function. And so why are snapping turtles vocalizing? Well, parental care is possible, but based on our knowledge of snapping turtles and their behavior, this is unlikely, and we don't consider this further in this species. More likely, it's possible that these vocalizations function in synchronized emergence or hatching. If this is true, then we may expect there to be an overall energetic or evolutionary benefit to nest sociality. And the reason for this is that less energy that is expended during emergence provides more energy for post-emergence activities such as escaping predation and finding a suitable site to overwinter. And so in general, this energetic benefit is called the social facilitation hypothesis. The goal of our second experiment was to see, in fact, whether nest sociality is energetically beneficial to hatchlings. And so to do this, we used a similar setup as the aquarium, except this time we buried the eggs in jars and covered the viewing window with Bristol board. More specifically, we set up a 2x2 factorial design where we manipulated depth and sociality. In the deep treatments, we buried eggs at a 5 or 20 centimeter depth. In the sociality treatments, we either placed eggs in the presence of 10 siblings or 1 egg and 9 plastic balls. And so the plastic balls were just used to keep the nest cavity intact. And then we recorded the day of first pipping and the amount of weight loss after emerging from the nest. And so in this talk specifically, I'm going to be focusing on the results that pertain to sociality, although there are results associated with depth. And so overall, we found that sociality, or the presence of siblings, significantly advanced pip date by 1.6 days. And pip date was determined, as seen in the picture, by checking each jar daily. And in the picture, you see a turtle pipping from the egg. And so that's how we measured pip date. And in the figure, we see that social treatments were often the first to pip with respect to each clutch. And so when we controlled for clutch, the pip date was earlier. And so also by subtracting the median egg mass before we place the eggs in their respective treatment from the median hatchling mass after they emerged, we see that hatchlings that emerged as a group lost 0.5 grams less weight than hatchlings emerging alone. And this may not seem like much, but this is approximately 6% of a hatchling's body mass. And so similar to the previous figure, average weight loss was significantly lower in the presence of siblings, despite differences in depth. And in the picture, you can see the hatchlings congregating just below the surface as they emerge as a group or they ascend as a group. And so what does this mean? Well, in short, it shows that sociality modifies hatch timing and provides an energetic benefit to hatchlings, where larger individuals are speculated to be more fit. And so the social facilitation hypothesis is accepted. However, this doesn't necessarily mean that vocalizations are a mechanism for this cooperative behavior. And so using our results from the first two experiments, we decided to directly test the synchronous hatching hypothesis. And we wanted to see whether hatchlings behaviorally respond to vocalizations. To test this component of the social facilitation hypothesis, we set up a playback experiment to see if vocalizations would affect hatch timing. To do so, we used a sample of 60 eggs from a total of 6 clutches and placed eggs in individual plastic containers to isolate any sibling interaction. We then placed each egg in one of the following treatments. And so in the picture, we see the MP3 player that we used to play back the sound and the individual containers with a single egg. These were also covered with plastic bags just to block out natural light. And so these treatments represent the sound that was played to each egg. And so the first treatment was a vocalization treatment where we continuously played 1 hour of vocalizations. And so this clip was obtained from the previously recorded pipping sequence, where we knew vocalizations would be played and a variety of types would be played in that sequence. And then the second treatment was a noise treatment where we continuously played white noise, which ranged from 500 hertz to 15 kilohertz. And this was the observed range in our vocalizations. And then the third treatment was a no noise or a silent treatment where we did not play any noise, any sound, and they in fact did not have an MP3 player. We then replicated each treatment twice, and we made sure to distribute each egg within each clutch. And then using an earphone splitter and earphones, we placed an earbud next to each hatchling and played the sound clips on a continuous loop. Then, and so each MP3 player was connected to 10 eggs, and then that was a total of 60 eggs. And so do vocalizations cue hatching? Well, after controlling for clutch effects and the egg's relative position in the nest, so that's the egg number, our results indicate that vocalizations do not affect hatch timing. In our figure, we see that the relative pip date is quite spread out over a period of 15 days, and the predicted pip date is quite similar among vocal white noise and silent treatments. And so going back to our hypotheses, even though sociality affects hatching and is energetically beneficial, it's unlikely that those behaviors occur through vocalizations. Other mechanisms for synchronous hatching could be synchronous or circadian rhythms, oxygen, which that includes oxygen consumption, metabolic rate, heart rate, or even another cue could be vibrations from the movements of siblings at hatching. And so this leaves us with the synchronous emergence hypothesis that has yet to be tested in turtles at large. And so since more complex vocalizations occur after hatching, it's possible that they facilitate nest emergence. The complex vocalizations that occur may indicate that more complex information is being exchanged, and in particular, relatively high frequency and short sound duration, which is what we observed, indicate that the sound is likely to function at short proximity. But we can't confirm this with our study alone. And so in summary, our study is, to our knowledge, the first to record hatchling snapping turtle vocalizations and be able to associate these vocalizations to a particular hatching period. Not many hatching behavior experiments do associate vocalizations to hatching periods. So this also adds to that. The study also adds to our knowledge of turtle synchronous emergence behavior, where groups emergence is energetically beneficial, so the social facilitation hypothesis. We also didn't know that this particular species can modify development timing, and this leads us to wonder what mechanisms allow these hatchlings to synchronize hatching. And finally, this is the first study to use playback experiments to test the function of acoustic signals in turtle hatching behavior. To date, hatchling vocalization studies have been largely observational, so we also can't generalize our results to all turtles, where it's possible that vocalizations cue hatching in other species, but this just hasn't been tested. And so the next steps of this project, which I will be continuing through a master's thesis at U of T, is to analyze hatchling vocalizations across freshwater species, specifically species in the province of Ontario. So that's through behavior tests and lab experiments. And then I will characterize the nest emergence behavior of these species. And then by comparing nest emergence behavior and species life history traits, I aim to try to disentangle the function of these acoustic signaling systems. And so because of different life history traits, we may expect there to be a difference between large-bodied turtles and small-bodied turtles, where the need to cooperate and coordinate emergence may not be as important in small-bodied turtles. And so thank you everyone for listening, and I'd like to acknowledge that this work wouldn't be possible without the ongoing supervision and collaboration with Dr. Christina Davey and Dr. Niall Rawlinson. And thank you to the British Ecological Society for hosting a great conference, and I look forward to your questions.